record. And we're recording.
Got it. Is my screen showing properly?
It is. You look great.
Awesome. Well, a lot of people, when they get into fungi, get into mushrooms in particular, there's a lot of talk about ecological roles, all the things fungi do in our environment, the roles they play, how they affect all the other organisms out there, and things really often tend to get lumped into like three major roles are really what we tend to hear about. You get, saprobic or saprophytic fungi that are considered decomposers. Some of the really common ones are litter lovers or wood lovers. They break things down. Then you have mycorrhizal fungi, which is a huge fraction of the delectable mushrooms people tend to go out and harvest. Associations with tree roots, or even other like Ericaceae, interesting relationships there. And then really this kind of garbage pail category of parasitic. And we've really shoved a lot of things into the parasite category. Generally, when it comes to fungi that people eat on a regular basis, that's largely plant parasites. But when it comes to the world of other things fungi manage to eat, how they manage to eat them, it gets really, really complex. You're dealing with the very fuzzy border between predation and parasitism. And really, I think there's one example that a lot of people have heard about, a lot of people have seen, and that's the fungal snares or nooses that some species will use to catch nematodes. And predation on nematodes is really, I think, what most people have heard about if they've heard about any of this. It's one of the most active forms and usually the least debated form of predation in fungi. One of the biggest aspects, predators, we think of like lions, sharks, hawks. These are things that run around, they seek prey, they have adaptations to capture, kill, and consume what they run into. It's like a clear case of predatory ecological roles, very clear cut. Gets a little messier when you look at things that eat immotile organisms. When you have a species that eats like barnacles, for example, it's an animal eating an animal, but because one of them doesn't move, researchers start applying labels like grazing. And you see the same thing with like sponges or any number of immotile like prey organisms. If both parties aren't running around, then the concept of predation starts to get messy, even just with animals. Fungi present a really unique problem in that they're completely immotile. There are a few fungi that produce moving spores, but none of them are really vegetative. The vast majority of fungi don't move at all, which is why applying the concept of predatory to them has been such a nightmare. Which is why this Arthropods noose has been such a great example. It really allowed researchers to start applying the term predatory to fungi. And the history on this actually goes back a really long way. So this is an example of a nematode, ensnared in a fungal noose. And the way these work is you actually have a ring of 3 cells. In a relaxed state, they're just a loop, not doing anything in particular. When they get triggered by mechanical stimulus, especially from the inside, they inflate in well less than a second, so fast that for almost 100 years, we didn't have the equipment to actually see what was changing, even see the dynamics allowing for this rapid inflation. But the general trapping concept is when a nematode pokes its head through, the noose inflates and traps it. The nematode's incapable of moving either way. Sometimes the pressure is great enough to just instantly incapacitate the nematode. And from there, you essentially get this hyphal peg that punches into the nematode. Within just a few hours of sensing that prey is there, it will punch into the nematode and branch until it fills the whole thing out and digests it. Now, This was actually recorded for the first time in, I believe it was 1880 by Zoff. He was the first person who actually recognized what was going on here. There was a couple really early on documentations of these loop-like structures, but he was the first one to actually note that it was predation going on. And he was the first one to actually sit there and observe both parties interacting to notice that this is going on. And after that was published, other researchers finally started picking up on the idea of predation in fungi. They started keeping their eyes out for other ways that fungi were interacting with animals, to try and see what more was going on. Because really, any scenario where a fungus was killing something else was basically always just considered parasitism because they were immotile. This is the first time we had really seen something like traps or really organized structures. And this species, Arthropods oligospora, was the first one described in the Zopf paper, but most of the Arthropods do this, can completely sustain itself only on trapped nematodes. Like, it is capable of trapping all of its food using this method. This trapping method, come to find out way later on, actually goes back a very long time. You know, essentially, as long as nematodes have been around, separate lineages of fungi have been developing the snare method of catching them. This image here is actually a predaceous fungi in amber, from about 100 million years ago. Now, it gets difficult much further past that. You know, amber is a great example of like fungal diversity trapped in time. But fossil fungi are terrible. We don't have many good methods. And the same goes for the prey in this case, nematodes. So our really best confirmed speculation with phylogenetics really estimates that nematodes probably about 400 million years ago, and it's likely that these snare producing fungi showed up right after. And that really leads into like kind of how it started. These snares are probably the best known and least debated example of active predation in fungi. There's very little debate at this point over calling that a form of predation. But what's even more common, what more fungi have actually used, and what likely goes back even further than nematodes, all the way back to when fungi first entered terrestrial habitats, is a different form of trapping, using adhesive knobs. It's been difficult tracking down images of a lot of these original relationships that are not sketches, other than Dreschler's, which I'll get into that. But the adhesive knob strategy basically consists of a small little peg or projection, which you can see these little appendages in the image on the right. They'll have a glob of adhesive on the end of that, and they can actually specialize the adhesive in that gel to target specific organisms. So the snares are essentially designed only for nematodes to wiggle through and get caught up. But these adhesive knobs could be specialized for essentially every other form of microorganism that they could run into, every other microanimal. So on the left is actually a dactylella that specializes in hunting amoebas, using adhesive strategies. On the right, you have images of both rotifers and tardigrades, water bears, getting preyed upon using this adhesive method. And even though the snares have really gotten a lot of publicity, these adhesive methods have really shown up more times, still on land, eating things. And a lot of researchers believe trapping of things like amoeba or other protists was probably one of the major strategies at the time. There's certainly a lot of evidence for it. And these adhesive knobs have been re-evolved time over time in a number of different groups. But this is a great example of a nematode also being caught by those adhesive knobs. You can see on the bottom that sort of knob with kind of a surrounding halo. That's the adhesive on the tip. And then what you're looking at is a nematode that's in contact with two adhesive knobs. And you can see almost how it has like a case. Some of these adhesives are strong enough where the nematodes will, you're looking at a nematode that is hundreds of times bigger than the two pegs holding it down. Like the power of these adhesives is insane. It's almost the equivalent of like a glue trap holding down an elephant long enough to digest it. But this is another example. Some of these species will produce not adhesive pegs, but actually adhesive spores that will break off if a nematode touches them. The nematode can then carry those further away. At a certain point, those spores will germinate, push a peg into the nematode, and it really just continues moving forward. You know, some of these not only was that observed, it was utilized. I guess that gets into a mycologist named Dreschler, who had an insane career, about 50 years of uninterrupted mycological research. He probably described most of the modern predatory fungal genera, and certainly many of the species. He's got a bunch named after him. But what he really pioneered was use of these nematode-eating species in agriculture. You know, these nematodes are mostly microscopic little worms, but they're capable of completely wiping out many different types of crop all over the world. Nematodes have bothered us as long as agriculture has been around. And Dreschler not only spent his time doing beautiful illustrations of some of these species, but trying to get them in use in agriculture with success. It created kind of this context of, can we find better nematode hunting species? Can we breed better nematode hunting species? Like, what all can we gain from using them? And eventually, what he discovered was actually a very unique species at the time that he named nematode-trapping. Up until that point, all nematode-trapping fungi had been either ascomycetes or zygomycetes or unknowns, effectively not fungi we generally know to produce anything. For the most part, they're not producing any large mushroom-like fruiting bodies. They're things we'd often consider molds. You know, they stay as mycelium. But he discovered this nematode isolate that was given to him by someone who's collecting like farm soil samples. He discovered it had clamps, which is a distinctive feature of basidiomycetes on their mycelium. It'll just be these little knob-like structures, and essentially that it suggested like, oh, wow, there might actually be that. And after these theories came about, you know, that was about, I think it was 1944 that he reported nematode. And then in 1970, someone actually found an isolate of nematode, tiny little mushroom. They handed it around to a bunch of other mycologists, and it turned out that what everybody had been calling nematode since Dreschler was actually Hohenboehlia, what a lot of people call the flower pot oysterling. It's a really, you know, this is just an example of a Hohenboehlia that we tend to see in throughout this region. And so mycologists had basically been reporting nematode for about half a century without knowing what they were. You know, they were publishing papers on mycelium, essentially, nematode eating mycelium. And at the same time, researchers were discovering and reporting on Hohenboehlia, and come to find out they were just looking at it from both ends of the same area. Like one person was describing the mycelium, one person was describing the mushroom. Once it got put together, come to find out every single Hohenboehlia that they've tested has nematode eating mycelium. They all do it. Which is also why I've been suggesting people, if they find, these little oysterlings in their flower pot or in their mulch, add that to your garden if you're ever having nematode problems. Nematodes pulling themselves out of their skin, I guess. But this was really the first mushroom that was ever linked to a nematode eating stage that had a predatory lifestyle under the surface. As time went on, a bunch of other groups were discovered. To later on, oyster mushrooms were actually discovered to be eating nematodes, but using a completely different strategy.
Hey, Sidney. I don't know when you're going to hear me.
Will actually have these big right now.
Hey, Sidney. I think we're having connection issues. It might be better if we turn off our videos, and then hopefully it'll use up a little bit less bandwidth, or however the internet works. Because there's a bit of what you're saying that I feel like are really cool points that we're missing. So we could try that.
All right. Is that helping at all? Can you hear me?
We can hear you. I'll let
Okay.
you know if it still remains an issue.
Perfect. Well, I'm not sure what you heard last. This strategy was discovered in a bunch of different mushrooms. It really started with Hohenboehlia, but from there, another close relative of Hohenboehlia, oysters. Basically, every species of Pleurotus that we've looked into so far has nematode targeting toxins produced all over its mycelia. These specialized droplets intended to stop nematodes in their tracks, paralyze them long enough for mycelium to creep up and actually start digesting them. At that time, it was only Hohenboehlia and Pleurotus that were known to eat nematodes. Out of all other mushrooms, no one had ever found any others that used this strategy. And at the time, the family that a lot of other species were lumped into, the Tricholomataceae, it was really kind of a mess. There was 5 well-known mycologists that had all published different versions of how they thought the family should be split up. Some of them wanted polypores next to Pleurotus. Some of them wanted Hohenboehlia next to polypores. It was all over the place. A researcher came in and actually pointed out, let me make sure I get their names right. It was Thorne and Barron basically came in and said, look, they share an ecological role. They share the strategy that nobody else has. We really feel like that should put these two close together. There's nobody else doing this. It would make a lot of sense if they might share a common relative. They spent a lot of time digging through the microscopic features of both species myceliums. They found a lot of intermediate features, and they were trying to put this theory together when, sort of similar to the first time someone just randomly had a culture of the nematoctonus that fruited. They actually got sent a sample from, and also from farm soil in Alberta that produced both nematotoxic droplets and the glue traps that you see in Hohenboehlia. it was a species that had intermediates of both. It really tied their theories together on this coevolution, that they share this trait, they belong together. And then they actually went through with some of these, at that point, relatively new sequencing abilities. They showed that, yes, they were closely related, but even cooler, they showed that this Hohenboehlia that had both toxins and glue traps was basal to both. So essentially, both Pleurotus and Hohenboehlia, as we know it, radiated out from this one that had both. From there, Pleurotus seemed to prioritize eating wood and nematodes kind of as a side game. But when you look at Hohenboehlia, it's a nightmare. You know, you have some of these that are known to produce fruit bodies. DNA has pushed a lot of these species together. What we found was that there's a lot of Hohenboehlia that only exist as mycelium. They never fruit, and they're hyperparasites. They only live as a spore spreading from nematode to nematode. They never really build these big, expensive mycelial masses. They only put enough energy in to spread glue-covered spores from nematode to nematode. And that's kind of a good example where if you can find something that would often be considered parasitic, if you can find another Hohenboehlia that is producing both glue-covered spores and mycelium, and then you can find another Hohenboehlia that's producing mushrooms, mycelia, and glue traps, which one do you call predatory? People had felt comfortable calling these highly adapted ones predatory for a long time, but when it gets into that parasite predator relationship, it really turns into a mess. Trying to apply any concept of animalian predation to species like this, I think fungi are basically a unique category. I don't think we'll ever fully be able to apply a concept like that to them. And really, I think anybody who's arguing that species like Hohenboehlia are anything but predatory should probably read more into the history and how much time other people have spent fighting about it. And it's done. We've really only found this and again and again. This is a really cool example. This is actually called a stephanocyte produced by a range of species, but mainly Hyphoderma. And they're actually essentially little backward facing spikes on a little knob that's coated in nematode attractive chemicals. So nematodes will actually come along, try to consume mycelium, which there's a lot of nematodes that prey on fungi this way, and essentially get stuck. It'll try to eat one of these spike covered knobs. It'll get stuck in its mouth and eventually grow out from the inside. Some of these will expand once they're in the nematode's mouth. Some of them are incredibly toxic as well as being like physically spiked. And the mace on the right was just a comparison by the author who did this illustration. And the SEM images are all terrible quality. So this illustration was really the best I could find, but we're really only finding more and more methods. One of the coolest was actually, these were dubbed spiky balls by the authors who discovered. These are actually produced by Coprinus comatus, you know, the well-known shaggy mane mushroom, preys on nematodes using these spike covered. It will specifically only produce these in the presence of nematodes when it needs a nutritional boost. Essentially, nematodes will come into and potentially through both toxin and physical action will basically get torn to pieces. And from there, you'll get other bits of the mycelium, little reaching out to mop up the remnants. Probably the most dramatic example of that strategy, though, is actually the acanthocytes that you'll see produced by Stropharia. And they'd been recorded for a long time. We knew these structures were there in most Stropharia, But in particular, there's a species, Stropharia rugoso annulata, the wine cap or garden giant. It's a really popular kind of wood chip bed cultivated species. And its mycelium is just covered in these acanthocytes, absolutely coated. And it took a long time for researchers to actually catch up and propose that they might have a function. On further analysis, the acanthocytes are also capable of just exterminating all nematodes within an area. This SEM image came out way after some of the original on their function were proposed. And after SEM analysis was really able to get a good look, these things function as hypodermic needles. You'll actually get these basically spike covered balls, a nematode against it. And not only will it get impaled, it will be poisoned at the same time, effectively injecting it with toxins to slow it down. And really, a lot of these have been noted in the past, like observations here and there. It wasn't until researchers like Dreschler early on basically started just watching mycelium, just seeing what mycelium did with organisms around it, watching like what actually happened when you allowed mycelium to interact with the environment like it would in the wild. One of the most revolutionary things about Dreschler's work that is outrageously simple, he started isolating these by putting leaf litter on agar and just letting it get contaminated, letting things grow and get crazy and wild, let things like nematodes and mites show up, and come to find out mycelium hunts, defends itself, like really all these different strategies of interacting that we had never seen. Like, there's really no way to shove so many fungi to just the three common roles we usually think about. Like saprophytic, mycorrhizal, and parasitic really cannot sum it up when you have species like this. And it doesn't even cover the range of others that will do things like just poison nematodes to keep them away or attract beneficial nematodes. It's really just, it gets incredibly diverse. The deeper you look, the more is going on. And a lot of this is super simple to investigate as well. Like anybody who has a microscope and agar setup can really start seeing what mycelium does. That was one of my favorite things to do not long after I started at college, was just watching mycelium. I can guarantee you the biggest reason we don't have more described predatory mushrooms is just because nobody's ever looked at their mycelium to see what's going on. The amount of strategies is really intense. And really, I just was kind of floored by how far back this went. Like seeing Zops publish on that strategy, basically get picked up, blow everybody's minds, and then have researchers go on for 100 years seeing all these different things going on, and it's still picking up to this day. Really phenomenal. I was very interested to read about a lot of this. But I think that basically covers the majority of what I wanted to go into without talking about how much people fought over Tricholomataceae being sorted out, or exactly whether something is poisonous if the glue causes it to be paralyzed before it dies. The details are kind of endless on this subject, but I suppose I'm basically happy to take any questions that are coming up at this stage. If anybody was wondering about any of this, I think that about sums up the main block.
So if anybody does want to ask questions, you can feel free to unmute yourself and just speak to us here on Zoom. If you're feeling shy or you've not confident in your connection, you can also put it in the chat and we can address those.
Let's see if I can switch back to video too.
Yeah, you should be able to. And you can, yeah, stop screen share. Perfect.
Awesome.
Hi.
I hope that internet connection wasn't too bad.
There was just a few parts there. There's a running transcript going, and I'm not sure if that, like trying to figure out your mycology terms, if it was just like breaking the internet a little bit.
It
Um,
usually does. And I wasn't sure how far I should go into like, how many people here know what a nematode is. People know exactly the mycelial dynamics going on. Like any question at all levels, like I'll answer anybody has really.
Okay, well, Roy, I have a question about, well, nematodes cause a lot of illnesses in humans. And I'm wondering if things like oyster mushrooms have, nematoxic, chemicals in them that might help deal with parasites in humans or animals. I mean,
Yeah,
nematodes.
absolutely, absolutely. So really what started the popularity was just Dreschler like basically proposing, oh, we could use these for crop nematodes. Later on in about like 80s and 90s, all kinds of strategies for managing human parasitic nematodes came up too. One of the great advantages to a lot of these predatory species is they're semi-aquatic or even fully aquatic. So there's like a number of human parasitic nematodes that either are primarily spread through water or have like crucial stages in the water. And there was great success specifically with some of the adhesive species, which those weren't mushroom forming. At least the papers I had seen weren't mushroom species. They were adhesive peg forming species used to basically trap all the nematodes in the water. And it's really so much more complicated than just like a glue trap that nematodes might wander into. Some of these traps literally produce nematode sex pheromones, or like nematodes produce specific smells when they're happy or they find a good meal, things like that. And some of these glue traps will pretend that they're a happy nematode or a nematode that wants to mate. And so they had great success pulling nematodes out of environments like that, human parasites, agricultural soils, wastewaters, a bunch of different settings. Oh man, so many good questions in the chat too. Did that answer your question, Roy?
Yeah, it was a perfect answer. Thank you so much.
Yeah, awesome. So I guess, yeah, let me pull this up. I'll try to go in order. So let's see.
I think the questions start with Fern. There's 4
Yeah.
questions there at the end, and the rest of it is just us troubleshooting.
Sure. So poor soils. Absolutely. One of the really interesting things about some of these predatory fungi, in the context of not only nematodes but other trapping too, is it's basically an on or off thing depending on whether they need it. Like the Caprinus, for example, the shaggy manes, if they're in a nutrient poor soil, they're having a hard time getting their nutrients. That's when they'll start trying to attract and kill nematodes. And like the first species I mentioned, the Arthropods oligospora, is able to survive in an incredible range of environments, many of them super nutrient deficient, because it can trap nematodes as a source of nutrition where other fungi wouldn't be able to. If most of the nitrogen in an area is running around as prey, exactly the same as like a bog environment, you won't have much of a choice but to try and trap that. And like Dreschler's work on finding and discovering new species, one of the simplest methods for that was basically starving fungi on agar to see if any of them started hunting, essentially. So, I mean, there are probably fungi capable of hunting nematodes pretty much everywhere, but they're certainly more likely to be found in a deficient environment. And if an area is chronically deficient, you might only see predatory species. I hope that answers your question.
Hi, Sidney.
Hey.
You cut out for a second, but I think you were saying something about how like you could put them in gardens possibly?
Yeah. Uh-huh. Probably. So a lot of these nematode trapping species are essentially existing as like what we'd call molds or just like mycelium. We'd never really see them in the environment. But probably the best nematode trapping species.
You're cutting out a little bit. You might want to turn
Yeah,
this
the Hohenwahelia. Um, we'll just stop video for a second. Probably the best one that you could use gardening would actually be the Hohenwahelia or the flower pot oysterling. They're essentially like, if you know what an oyster mushroom looks like, you'll basically see them growing on soil, just sort of these almost petal shaped mushrooms. They're really common in flower pots, really common in mulch or like really decaying logs. And every single one that they've ever looked at is just an insanely brutal nematode killer. The spores produced by that mushroom literally need nematodes to breed. A nematode has to pick up those spores, at least 2 different mushrooms, and get killed by them before they'll mate and reproduce. So really, if you just took those mushrooms that you found, the Hohenwahelia, like ground them up into water, like what people call a slurry, and dumped that around your garden, they'd almost certainly destroy nematode populations wherever you put them.
Would they also give you like fruiting bodies without messing with your vegetables, or no?
If you had a bunch of wood in your soil, you might see mushrooms popping up, but they wouldn't mess with your plants.
Wow.
You know, they're primarily either hunting nematodes or making the best of whatever mulch is around. But at all stages, they produce brutal nematode killing traps. And then really the other one people might try, people put them in gardens anyways, is garden giants. The Stropharia rugoso annulata, or wine caps. Their mycelium produces those spike covered structures that kill nematodes, probably not as efficiently as the oysterlings, but they produce huge delicious mushrooms. So a lot of people will have them in their gardens anyways, just as a source of food. They're easy to grow. So you might get a double whammy there.
Sweet. Sounds pretty
Yeah.
good.
Absolutely usable in a number of different contexts for agriculture.
Oh, Sidney, can I jump in? This has been a great talk. I've been trying to explain what you've been talking about tonight, but not as eloquently because I don't have the depth of knowledge, but just trying to explain to people this connection with all this mycelium and mycorrhizal based activity and how deep it goes. Like you were describing these traps in the glue and use of pheromones and things like that. It's just so beautiful. So thank you so much.
Yeah, absolutely. And now I will go on to one of the chat questions real quick. I don't want to neglect those. So it looks like someone missed my Hohenwahelia recommendation. I'd just basically get the mushrooms that you see, put them in a blender with some water. Tap is probably fine. And then I would just spread that anywhere around your garden, really. You know, those are probably the simplest methods they've ever been used in agriculture. But really, even prior to that, most of the nematode trapping species had been trialed in different agricultural contexts, often with great success. And then let's see. Are fungi known to predate on any other organisms? So nematode trapping fungi are probably the most well-known, the most well-studied, because they have had applications in agriculture and human health. But some species have actually even been recorded preying on things as large as springtails, if you're familiar with those. There's like, I guess this kind of goes back to the really poor environment question. But there's certain communities in the certain ecosystems in places like Antarctica where it's very cold. There's very little free nutrition. And you have these fungi that will kind of do a dual role thing of parasitizing and predating on springtails. That's probably one of the biggest things that you'll see them eating. And you know, for context, some of those are like 2 millimeters long. But then really, I don't think there are any micro animals that haven't been observed falling for some of these glue traps, like amoeba, tardigrades, water bears, mites, rotifers, really anything that is carrying around nitrogen and has an appendage that can stick to glue is a vulnerable organism to some of these fungi. Really, when it comes to things like fungi eating animals, like vertebrates, for example, that's when it gets a lot more contentious. You know, it's already hard enough to say if a glue trap fungi is truly a predator, or if it's just kind of like passively throwing out traps. But when you have a fungus like the white mildew that's eating bats all over the US, for example, that cause white nose syndrome, that's a species that is not trying to parasitically coexist. It's not aiming for long-term cohabitation with the host. It's literally eating bats alive, spreading from bat to bat. It can vector around in other places, but I think there are a number of examples like that where typically those would be called parasitic relationships. But if the goal is truly not even slightly to attempt to coexist, some of these parasites, like the second they attach to their host, are pumping out toxins to kill it as fast as they can. There's things that try to do that to people. And I think bias in relation to the size of the thing trying to eat you and the size of the thing being eaten has also contributed. But in my radical opinion, I think things like white nose syndrome are arguably predation, and that we need to give fungi a little slack because they can't walk around.
Yeah.
But then, of course, you could argue a lot of bacteria are predators. Then it's messy. This is not clean cut. Fungi manage to bend basically every rule we try to apply. And so let's see. Coevolutionary strategies. That's a great question. There's a lot of there's basically nematodes for all ecological roles. Like, you know, similar to fungi, there's nematodes that have worked themselves in everywhere. And there are a lot of nematodes that in particular will target fungi. So they're basically fungivorous nematodes. They have special mouth parts designed to like suck the contents out of hyphae. Some of those species have adapted around predatory fungi. Either they're resistant to toxins put out by species like Pleurotus, resistant to, like some species of mushroom actually have basically poisonous mycelium to nematodes. Some of the nematodes have adapted around that. And really, when it comes to, like, I'm trying to think, you know, there's been some weird examples. I'd have to do more reading on that subject, honestly, to give you a better example. Really, the resistance to toxins, avoidance of some of the lures and glue. But when it comes to like crazy anatomical adaptations, I'm not quite sure. Some nematodes are just too big to be trapped, but I don't know if that would count. And then I guess, was there any audio questions before I do more chat questions?
I have a quick question. Do we know how long it takes the fungi to break down or digest the nematode? Like, is it for like a really long period of time, or is it quite quick?
Oh, it's incredibly fast for some species. So with some of the arthropods, which are, most of them are like the snare formers, they're already penetrated and branching inside the nematode in just a couple hours. And usually by 24 hours, there's nothing left but mycelium. For the Stropharia, the garden giants, which produce those crazy spikes, oftentimes the nematodes will be shredded into like pulp essentially. And then that just like the mycelium absorbs that almost immediately. The researchers actually noticed that they would observe nematodes turning into clouds and then disappearing, or just like smears of nematodes. They saw nematodes like die of their injuries and dissolve. There's all these different ways that they want to take advantage of it. Because I guess it's sort of like a Venus flytrap will close to hold off that nitrogen that it's captured from everything else. A pitcher plant will have anatomy that bogarts that nitrogen for itself. But when you have something like a fungus that can't really do too much to try and like drag its prey up a tree, many of these nematode eating fungi will incredibly quickly break down the nematodes. Like under a day is probably average for most of them, but typically you see that first like peg, that first punch in under a couple hours. If that answers the question.
It does. Thank you.
Yeah, absolutely. Was there any other audio questions? All right, I will get to the adhesive substance question. It's a mix of proteins and carbohydrates. I actually had a hard time finding good published data on composition for a lot of the species that like Dreschler described. But for some of the ones that specialize, like when you were looking at ones using glue traps for amoeba versus glue traps for nematodes, there is protein specialization in that adhesive to like preferentially bind to whatever they're trying to make contact with. So like one really interesting thing about the glue traps for amoeba is, amoeba, they're blobby, they don't have a cell wall or anything. The glue trap will basically sink itself inside the amoeba, and normally an amoeba would never have a hard time pulling away from something solid that it was trapped up on. The glue on the nematode, on the amoeba traps, basically ties up all the crucial parts of the amoeba's anatomy, so it can't go anywhere. And with like the Hohenboehlia, those are basically nematode skin specific proteins. And that's why like numerous researchers observed nematodes like either tearing portions of themselves off or like separating from their skin, because just immediate brief contact will have like irreversible adherence. And that's mostly because of specialized proteins. But I know there's carbohydrates involved. I believe it's mostly proteins that are doing the irreversible cementing though. And then next question, molecular switch, context dependent. So for like oysters, for example, Perotis, they don't necessarily always put the energy into producing nematocidal compounds. They're not always trying to parasitize or like prey on, just murder nematodes in their presence. And that's largely dependent on how much nitrogen they're getting. So in that case, it'd be like, if I start to need nitrogen, that's what's going to switch on production of these structures. There's similar things like with the Caprinus, what they saw is you could induce it two ways. You could get their so-called spiky balls to form either by depriving them of nitrogen or exposing them to the presence of nematodes. I don't believe they cited a specific molecule indicating there, but it's almost definitely some form of metabolite, waste, or pheromone produced by active nematodes that's triggering that. And certainly when it comes to like the closer to classical parasite like Hohenboehlia, they'll really aim their structures towards other nematodes. So presumably, again, some nematode metabolite or pheromone. But when it comes to like Hohenboehlia, they're usually always producing these structures. So you can basically always put them somewhere and you're always going to have glue available. That's probably the only reason anybody cared to describe so many before we ever connected them to the mushroom. But that nutrient enrichment was a big problem for like arthropods, actually. Like that first one that forms snares. It might not try to form many snares if it's a really rich soil. It's kind of a bad example because arthropods also does things like enter root tips and parasitize other fungi. So kind of a weird example, but that is one where you might have to worry about what your soil looked like if you tried to use it. And same with like Pagetospora, or wait, is it? It's either Pagetospora or Pagetomyces. That's another Dreschler species. Or like Dactylella, a lot of those non-Basidiomycete, not necessarily obligate nematode hunters will give mixed activity.
Can I ask another question?
Yeah.
You do a lot about, I know you focus a lot on fungal reproduction. I just wondered how you, what interested you about like fungal predation and kind of got you engaged in this subject for the talk.
Well, one of the first things I stumbled upon was the Barron, I think it was Barron and Thorne. I have all my citations on a paper. Was the Barron and Thorne paper essentially like rectifying the mess of a family that Tricholomataceae was? They came at it from a really interesting angle. They used, they were basically going at it from like a morphology angle. They had your classic like bread and butter microscopy to support it, which is mostly what everyone was fighting about at the time. They were working on getting the genetics together. And then what nobody else did is they tied in the behavioral aspect. They looked at a shared like ecological behavior that no one else, none of the other mycologists at that time were even thinking about. And they really had a solid idea with that pattern of behavior. And then when they went back and actually, like, I guess almost serendipitously found that species that shared both traits, sequenced it, and it turned out to be basil. What it makes me think about is how you have that first Hohenboehlia producing both toxins and glue traps, basically foundational to both strategies later on down the line of species that only do toxin or only do traps. And then you just look at all the different ways that the Hohenboehlia have diverged. You know, there's Hohenboehlia that produce big mushrooms and big mycelia and have like interesting behavior that they built out of this basal trapping. And then there's other ones that have gone the other direction. They've abandoned large mycelia. They don't ever really produce fruit bodies, or at least nobody's ever seen them. And they're basically spreading primarily as tiny glue covered spores because they've managed to specialize the strategy well enough to get away with it. So when you have an example like this, where like you look at the starting material, which just presumably something similar to this Hohenboehlia that had both, and then seeing how it diverged out to form like two big groups with such, like, I guess what I really like there is seeing the evolution of behavior represented in Hohenboehlia specifically. That gets me thinking about reproduction, and like, it really also kind of blows my mind when you go back and look at the data and see that those nooses showed up basically as soon as nematodes did. Like fungi were ready to start specializing in this. And that gets into the breeding and genetic change that we can see in fungi so damn quick.
That's pretty great. Do you
Yeah,
see any,
I think that's a great
did
example.
you see evidence of any cooperative behavior when it comes to like an ecological perspective in relation to nematodes anywhere else? Or is it all essentially what you've seen predatory behavior around nematodes?
Well, when it comes to like arthropods, for example, which does weird things like punch into root tips, you would get like selectivity for plant pathogenic nematodes. So you might have an arthropods that's benefiting more by punching into plant roots. Well, not punching into, wrapping around plant roots, entering nematode-filled galls, clearing them out. And it can get a bigger payoff that way than it is expanding around the plant and evacuating the soil of everything indiscriminately, which is kind of what they'd normally do. So that's probably a good example where you see like interesting community changes due to some of this behavior, where there's a little bit of a weird trade-off going on where it's going to help out the plant because it ends up working out a little bit more. And the other nematodes, beneficial nematodes, are having a better time because of it. But it's also really hard to generalize. I mean, some of the like really, really obligate predators end up targeting just like one or two species, you know, like the noose or snare formers. They're just size discriminate. Any species of nematode that gets caught in that is really gonna go. But for some of the others, they really end up specializing in like one species, and that might result in kind of a cascading community change.
Yeah, that's super great. Thank you.
Yeah, no, great questions. And I think that also kind of answers Adrian Keys's question too. So let's see. I'll get to a couple of chat questions. Question from Melinda K regarding saprophytic species using these strategies. It really gets into a lot of mycorrhizal species have weird nitrogen obtaining methods. And when it gets into species that have things just like poisonous mycelium or metabolites that kind of happen to murder all life in an area, like Lacaria, for example, this was a while ago, so I can't quote my source, but I believe that they were one of the species that did things like nuke all the bacteria in their area and just sap the nitrogen. That's the type of behavior you'll see at a mycorrhizal species. And I don't know that you necessarily see things like really, really specialized nematode toxins like in Pleurotus, or really organized structures like nooses or acanthocytes. But honestly, the mycelium of mycorrhizal species is often even more atypical than just the weirdos you see in soil. I'm not actually sure that we've looked enough, but I also, in my literature search, didn't see Lacaria. I didn't search Lacaria or many mycorrhizals. So I feel like I can't fully answer the question. Hope that's okay. Let's see. Yeah, that actually is a great point on white nose. From what I understand, it's really species specific. But when you get into, what is it? Much better with fungi than bats. I want to say it was the little brown bat, right? Where the mortality rates in infected bats are close to 100%. But if you look at a species that's making the transition from parasitic to predatory, for example, I'd argue that what's a good example? I don't think I'm going to have a great example for that, but that would really be my idea there. Like, I really feel like we push parasitic on too many fungi in too many contexts. It gets messy when you look at a lot of different examples, though, like with bacteria too. Where is the line on predation versus parasitism? Is there size bias in how you're judging it? And there's a lot of predators that prey manage to coexist with. If survival rate of an infection with white nose in one species is 5%. And the success rate and the attack and kill success rate of something like a shrew on grasshoppers is also 5%. Could you draw comparisons there? Like, I don't think there's many predators that are 100% successful on any population they're targeting, but not all predators are attached to their prey. I'm making some serious leaps here, I realize.
That answered, I guess, the question that I had really well. And it also brought to mind, and I cannot remember for the life of me. Oh, actually, yes, I can. The chytrids, which I know are kind of, right, are they kind of fungus, but not quite. But to speak to that point, you know, the chytrid fungi or whatever they are, it seems like they're predating on the amphibians all across the world and causing a really big problem, similar to the white nose bat or the white nose fungus for the bats. But it just kind of drew parallels for me while we were talking. So that makes a little bit more sense what you were saying in terms of predation for those. So thank you for that.
Yeah, I think that's another good example. And chytrids are also really, really weird. You know, they're one of those fungi or basal, very basal, that actually do have a moving stage. It's not a vegetative moving stage, but they do weird stuff. It'd be hard to draw a lot of comparisons between them and other fungi, but certainly the effect they're having is comparable to white nose. Maybe even worse in terms of the range of species affected. Let's see. Well, we're almost out of chat questions.
Robert wants to hear the good news about nematodes. There's been 2 comments that are like, do we really want to kill them? Aren't they okay?
Well, I mean, I think there's a lot of people who would enjoy killing off the parasitic nematodes. And certainly when it comes to human parasitic nematodes, I think there's some arguments to be made. But, you know, nematodes, there was a great quote, and I'm under-researched on nematodes themselves, absent the context of fungi. But it was along the lines of if you just made all surfaces on Earth invisible, you know, everything except for nematodes, there would just be a thin film of white covering everything, because nematodes have effectively worked their way into every niche. They're coating all surfaces at all times, doing essentially everything, an enormous fraction of our biomass. So we certainly would not have a good time if we killed all the nematodes. Well, yeah, unless there are any other audio questions, I think I hope I got to everybody in the chat. And I hope everybody enjoyed the talk. Hope I didn't ramble too much and audio worked out.
Can
Totally
you
great
throw your face up
presentation.
there one more time?
Yeah, we'll see if it murders the audio.
Thanks a bunch, Sid, and thank you, Humboldt, for hosting this.
Oh, we are so happy that Sidney agreed to do this talk. And thank you for coming.
I am very glad for the opportunity. This was a great talk to cover, and you know, so much to fit into one thing. I was grateful for the opportunity, and really some great questions. I didn't expect like really that range. It gave me the opportunities to talk about a lot of the stuff I didn't get to include in the chat. So thank everybody who attended, and thanks to the society, and I had a great time.
All right, and so we'll post this recording to the Facebook event, and a link to it will go out in our newsletter. And Sidney, if at any time you would like the recording to be removed from the internet, you just let us know, and you're welcome to share it with whomever and do whatever you want with it, since it's your intellectual property.
There's definitely a few people who wanted to get it, so I'll look out for the link. Thank you.
Sure.
Awesome.
Thank you for hosting, and thank you, Sidney.
Yeah. Bye-bye, everybody.
Bye.
Well, we still had pretty good attendance for not being on our regular night.
We absolutely did. I was checking, we had a lot of people there.
Yeah, a lot of people paying attention and psyched too. Thanks everybody for all your great questions.
Alrighty.
Yeah, I guess I'll wrap it up. I can stop recording. That's something I could do.
Without further ado, I want to leave.